Hello, grade 8 students. In this session, we're going to continue with our chapter Digestion, part 6. Let's talk about the enzymes. Enzymes, they are the agents of digestion. If you remember, enzymes, they are the biological scissors that cuts, break down, digest, hydrolyze the food into smaller pieces. Also, they accelerate the digestion without being consumed at the end of the reaction. Today, we are going to determine the conditions needed for the proper functioning of the enzymes. So, in this application, we're going to talk about the experiment. We're going to interpret. The meaning of interpret: analyze plus an indication. So this is the interpret. And also we are going to deduce the enzymatic property in each of the performed experiments. And here we go. In this experiment, we have to do this experiment to demonstrate a property of an enzyme, the specificity. The enzyme is specific. Here, in this experiment, this is the goal, or this is the aim, or the purpose, or the objective. So what is the objective of this experiment? Is to determine the property of the enzyme, which is the specificity. Here we have, in this experiment, we have the conditions in figure A, and in figure B, we have the results obtained after 1 hour. So here, in this experiment, we have two test tubes. They are placed in a water bath where the temperature is 37 degrees Celsius. It's the normal body temperature. In tube A, we cooked starch plus the enzyme. No, it's the saliva, not the enzyme. Saliva contains the enzyme. So don't mix. You don't have to say the enzyme. You have to say the saliva. Saliva contains the enzyme that we call it the salivary amylase. It works on what? It works on starch. To give what? To give maltose, which is a reducing sugar. So this is the importance of the table that I told you. You have to study it about the enzymes. So in tube A, cooked starch plus fresh saliva. We said that saliva is not the enzyme. Pay attention to this. While the saliva is the juice that contains the salivary amylase that works on starch to give maltose, which is a reducing sugar. While in tube B, we have albumin of an egg. So albumin of an egg, we talked about this the last time, it's protein. So albumin of an egg, it's a protein. So plus fresh saliva. Results obtained after about 1 hour. In tube A, we did the Fehling test. For what do we do Fehling test? For reducing sugar. So we did the Fehling test. What happened? The Fehling test, we have brick red precipitate. What does this mean? This means that we have reducing sugar. So what is this? We have starch. So what happened for the starch? It was digested by the salivary amylase in the saliva to become reducing sugar, which is the maltose. While in tube B, albumin of an egg is protein. After 1 hour, we did the Biret test. So it gives a positive result. It gives a violet color. This means that we have proteins and or polypeptide. So this means that albumin is not digested by the enzyme that's found in the fresh saliva. So here, let's interpret. So the variable factor here you have to know that is the substrate. The first one, we have starch. In the second tube, we have albumin. So the variable factor is the substrate. So after adding fresh saliva to the cooked starch in tube A, the result we have brick red precipitate with Fehling. So this is the analysis. What does this indicate? This indicates the process of reducing sugar obtained from the digestion or from the transformation of starch in the presence of fresh saliva that contain the enzyme salivary amylase. However, after adding fresh saliva to albumin in tube B, so here's the variable factor. Here we have cooked starch, here we have albumin. So this is the variable factor. The result is that we have violet color with the biotid. So this indicates the presence of proteins and or polypeptides present and not transformed by the fresh saliva. So here we have digestion only in tube A and not in tube B. So the enzymatic property here, every enzyme works on a specific substrate. So this is the first property about the enzymes. Number one, every enzyme acts on a specific substrate. In this case, enzyme in the saliva, that's salivary amylase, could digest starch introducing sugar, which is the maltose, but could not digest the albumin. So this is the first property for the enzyme. Every enzyme works or acts on a specific substrate. Salivary amylase, it works, it digests only starch and not protein. Figure C. Activity of the enzyme and the pH of the medium. Acidic, basic, or neutral. C, for the pepsin enzyme, when the pH was 1, the activity was minimum. When it increases to about 2, the activity it increases to the maximum. And then the pH increases from 2 to about 3.8, 3.9. Here it decreases back to the minimum. This means that as the pH increases, it stimulates an increase in the speed of the reaction. So here pH, and we have the speed of the reaction. So as the pH increases, the pH of the reaction increases, the speed of the reaction increases, but up to a certain limit, to a specific value, which here for the pepsin, it's 2. While salivary amylase, as you know, it works in about a neutral medium. So when it was 5, here the activity was approximately null. When it increases to about 6.8, 6.6, 6.7, approximately neutral, it increases to the maximum. So the speed of the reaction increases to the maximum at this value 6.5 or 6.7. However, at the pH increases to about 9, the activity of the salivary amylase decreases back to the minimum. While for the trypsin enzyme, as the pH increases from 6.5 to about 8.5 or 8.6, which is here, it increases to the maximum speed, and then it decreases back to the minimum whenever it moves to the pH 9.8 or 10. So here we can know that pepsin, it works in an acidic, salivary in a neutral medium, whereas trypsin in basic medium. So the pepsin, trypsin, and salivary amylase, the variable factor in all of these is the pH medium. So for the pepsin, when the pH was 1 acidic, the speed was minimum. As it increases from 1 to 2, the speed increases from the minimum to the maximum. While as it increases from 2 to 3.8 approximately, so the speed decreases back to the minimum. This indicates that increase in the pH, it stimulates an increase in the speed of the reaction, but up to a certain limit. In this case, pH equal 2 was the best. Salivary amylase, when the pH was 5 acidic, the speed of the reaction was minimum. As it increases to the 6.5, approximately neutral, so here the speed increases to the maximum. And then, as the pH increases from 6.5 to approximately 9, which is basic, then the speed decreases back to the minimum. This indicates that increase in the pH stimulates an increase in the speed of the reaction up to a certain limit. For trypsin enzyme, also for pH equals 6 acidic, the speed was minimum. As it increases from 6 to 8.5 to the basic, the speed of the reaction increases from the minimum to the maximum. And then, as it increases from 8.5 to approximately 10, the speed decreases back to the minimum again. This indicates that increase in the pH stimulates an increase in the activity, but up to a certain limit. In the case of trypsin, 8.5 was the best, was the optimum pH, would mean that the best pH. Here, the second enzymatic property, every enzyme needs a specific chemical medium or proper pH. So till now we have two different properties. Every enzyme works on a specific substrate, and every enzyme needs a specific pH medium in order to digest and to work. Third figure, talking about the activity of the enzyme and the temperature. So as you see here, when the temperature was 0, the activity of the enzyme was 0%. This is the activity of the enzyme, and this is the temperature. As the temperature increases from 0 to about 37 here. So this is 37, this is the optimum temperature, and this temperature is the best temperature for the enzyme to work. The activity on the enzyme was 100. And then as temperature increases from 37 to 60, the activity decreases back to the minimum. So here, the variable factor is what? It's the temperature. Temperature was 0, the activity of the enzyme was 0. As temperature increases from 0 to 37, the activity increases from 0 to 100. And then this activity increases to 0 again when the temperature increases from 37 to 60 degrees Celsius. So this indicates that increase in temperature causes an increase in the activity of the enzyme, but up to a certain limit, which is 37 degrees Celsius. So the third enzymatic property, every enzyme needs a proper temperature, specific temperature to work. Mostly, in our case, it's 37 degrees Celsius. So to keep in mind, what is the optimum temperature in this case study? 37. Why? Because the enzymatic activity is at its best at this temperature. Before we said because it's the normal body temperature. From now on, you have to say this, since the enzymatic property is at its best. So the function of the enzyme is best at 37. The enzyme is going to be active at 37, while it's going to be inactive. Temperature is 0 degree. While because the enzymes, they are inactive, they can't work. The activity is going to be 0, so they're going to be freezed. So the enzyme is going to be inactive at temperature 0 degrees Celsius, while 60 degrees is the denaturation or the destruction. The enzyme will be destroyed at high temperature. Even though this temperature we gave it back to 37, this enzyme will not work because the enzyme is denatured or it's destroyed. So you have to know both denatured or destroyed. So as a conclusion here, if an enzyme was inactive at cold temperature and then we replace it into normal temperature with the 37, it will function again. While if the temperature was 60 degrees, let's say high temperature, in this case 60 or 70 or 80, and then we put it back to the normal temperature 37, it won't work. Why? Because the enzyme at high temperature will destroy or denature the enzyme. So the enzyme will be denatured. So these are the three important factors for the normal functioning of the enzymes and the notes in this session. Thank you guys. Bye bye.